Hi, good afternoon, everyone. So, we will begin today's session. Today, again, we will look at week five's content, kind of summarize the content, where mostly the content was all about proteins, right? We have been looking at biomolecules, carbohydrates, lipids, nucleic acids. So, this week's content was all about proteins. So, we will look at what proteins are, how to how to characterize it, what are its composition, like what are its constituents, and how to characterize proteins, etc. Like very briefly touch upon the things, and then look at few questions. Okay, so I will also in this section, like I had previously recorded one part of like almost the basics of protein biology, like amino acids and stuff. So that part I will play the video. Okay, so the recorded lecture only, and then like I will be here, it is recorded only, and then we'll continue with the remaining aspects that was not covered in that recorded part. Okay, so I will start sharing the screen. All right. So, in general, what proteins are? Proteins are basically polymers of amino acids. And amino acids are actually, the name comes from, it has two ionizable groups basically. One is this amino group, NH2, where it is capable of, accepting um, like about donating electrons, like giving it an overall positive charge. Or you also have this carboxylate group, COH, where it can actually donate H plus ions, thereby giving it CO minus an additional negative charge. So all these ionizable groups, along with the side chain that is there, the R group that is variable in case of the amino acids, these overall give a net charge on the amino acid. Each different types of R groups can eventually add to the overall net charge of an amino acid. And these each amino acid, we will look at it eventually. Okay, so these amino acids are joined by covalent bonds, which is known as peptide bond. Eventually happens between like NH2 group of one amino acid and COH group of the next amino acid, and eventually thereby dehydration takes place wherein one water molecule is lost, thereby forming this peptide bond. And each protein can be broken into the constituent amino acid by different kinds of methods that also we will look at eventually in the later slides to study the free amino acids. And there are 20 naturally found amino acids, um, in which comprise a protein sequence. Okay, so this is the video. I will play it fully. There's no audio? Oh, oh, array. Oh, oh. But you can hear me now? Oh, oh. Mm-hmm. Oh, oh. All right, maybe then I have to go one by one, that thing. Just a moment then. If I play this, my screen is visible, right, to you guys? All right. Let me play that video, the file. I had embedded it in the PowerPoint. So let me know if you are able to hear this video, the audio here in this case. Just can you hear this? Uh-oh. All right. Okay, wait. So maybe, all right. What I have to do? Still nothing? Okay, okay. All right. Hmm. The thing is, okay, wait. What I can do is just. What I can do is, because this is a video, right? So I don't have the slides of this video exactly right now. Later, like based on what amino acids are and stuff like that. So maybe I can touch upon, because I don't have my writing pad with me also. So what I can do is touch upon based on the slides shown in the video only. I will touch upon what all things are there and then look at the next slides. Okay. So just yeah. So in general, what we will be looking at is proteins. Uh, the very monomeric units are the amino acids. Okay. And here, and then the amino acids come together. Each amino acid is joined together by peptide bonds. And eventually, each amino acid peptide bonds, the primary sequence, it leads to a primary sequence. The consecutive monomeric units joined by peptide bonds forms a primary sequence. Eventually, that primary sequence folds onto itself and forms the structure, and eventually this how the structure eventually guides the function of the proteins that we see all around. Okay, so now if we look at the general structure of the amino acid, okay, so it has a C alpha. So what whichever C atom is joined to this particular carboxylate group, so this is called C alpha. Okay, and it has NH2 group here, it has COH group here, and one H atom bound to it, and this is the variable functional group. Now the simplest amino acid is glycine, in which case the R, the functional group in that case is H here. Okay, and so, yeah, in general, what I was talking in the first slide also, any amino acid has at least two ionizable groups, which are NH2 and COH. And based on this particular functional group can actually vary across the 20 amino acids. It can either be only H in case of glycine. Then you can also have positively charged residues here, giving rise to positively charged amino acids. We will look at each of them eventually. And or you can also have um, like negatively charged groups or polar functional groups attached in this position, okay, which eventually drives the uh, characteristics and affects the overall protein structure as well as its function. Okay. So when we talk about so we looked at stereoisomerism or chirality in case of carbohydrates as well. So here also in general, like in nature, we have L amino acids existing. L means L and D basically refers to L means levorotatory and D means dextrorotatory. Basically, depending on which way the plane polarized light is rotated by these molecules. When these molecules are present, they can either rotate it anti-clockwise or clockwise. Depending on that, this L and D forms come into picture. All right, and these, like this is the C here. This has COM, like carboxylate group COH or carboxylate. Like here it has been shown that it is existing as carboxylate ion, and this is as NH3 plus. Or and here this particular C alpha carbon has four different groups attached to it, right? Like here in case of like glycine is the simplest amino acid we looked at, the R group is H here, and next comes CH3, which is alanine here. The R group is CH3 here. So in general, this C alpha carbon, if we look at it, it has four different groups attached to it, eventually giving it chirality. Okay. So this has non-superimposable mirror images. Eventually, the carbon here is chiral carbon. Okay. So this is where the, in case of amino acids as well, you see stereochemistry in this case as well. So So that is what is, like I was writing here, that this is actually a chiral carbon, the star marked carbon, which is asymmetric due to four different groups attached to it. Okay. Yeah, this is levorotatory, this is dextrorotatory, and so this is the wedge and dash representation of any chemical compound, where you see these ones, like if you suppose this is present in a plane, so these representations represent atoms or groups coming out of the plane or going away from us, going to like coming towards us, projecting towards us, or projecting away from us. Okay, so this is called a wedge and dashed representation of chemical compounds, right? So yeah, so amino acids in proteins are L stereoisomers. Okay, naturally occurring ones are L stereoisomers. Now here, so in general, the 20 amino acids, if we look at the 20 amino acids, so we have the different R groups I was talking about. Here, the simplest one is glycine. Then you have alanine, proline, valine. These all the different R groups actually dictate which category these amino acids belong to. So the very first one is non-polar aliphatic R groups. So it has simple non-polar carbon chain attached with this C alpha form, like proline here, valine here, methionine. Is one of the, in general, you guys know that during protein translation, like protein synthesis, the very first amino acid that gets incorporated or that gets encoded is methionine. Okay, and methionine has a sulfur group attached to it, and then you have isoleucine and leucine as well, and then you have aromatic and another amino acid proline is present. So in case of proline, what happens is here you have free like free normally attached groups here. However, there you have a heterocyclic ring attached to this C alpha carbon. Okay. And eventually this actually affects the overall uh, sequence and the structure of the protein wherever proline is present. Okay. And another group comes aromatic R groups, where you have phenylalanine, tyrosine, and tryptophan. So these particular amino acids have like bulky benzene rings, or more complex like heterocyclic rings. However, it has double bonded character and eventually delocalized electron cloud all over, which gives it the aromaticity. And also in general, like we looked at nucleic acids, right? We can measure the absorbance of nucleic acids at a certain wavelength, which was 260 nanometers. Similarly, in case of proteins, especially these aromatic residues, due to the presence of these delocalized electron cloud all over, like around this heterocyclic chains. So here you can actually see, you can actually measure these particular aromatic amino acids can give absorbance at 280 nanometers wavelength. Okay, so you can eventually measure the concentration of proteins there. Then you also have polar uncharged polar uncharged amino acids as well. One here is serine, then another is threonine here. Then again, one was methionine having a sulfur atom there. Similarly, cysteine is there, where you again have a sulfhydryl group SH. Then you have asparagine and glutamine, which are polar residues because of the presence of these N atoms with lone pairs here. Okay, these two. And then the next comes positively charged R groups, where you eventually have uh, lysine, arginine, histidine. Uh, Lysine, because the overall you already have the NH2 attached to the C alpha carbon, and the R group eventually also has another NH2 group at the end. Okay. And similarly, you have arginine, which again has NH2 here, and histidine again has a heterocyclic ring here also. However, you also have an N atom which can actually donate its lone pair and eventually forming, like giving a net negative charge. And another one is negatively charged amino acids, where the R group is like again an additional carboxylate chain, where you have aspartic acid or aspartate and glutamic acid or glutamate. Okay. See if I'm missing something here. The same things are being repeated. One more thing. In this case, so we looked at asymmetric carbon in general for an amino acid is this C alpha carbon. However, there's like isoleucine is there, where you can see that the isoleucine here, so this is C alpha carbon, this is C beta carbon. Okay. And you can see that if you now see this particular beta carbon here, this is also asymmetric because you have one H atom attached to it. This particular beta carbon also has four different groups attached to it. Okay. One is this H atom here, one is this CH3, you have C2H5, and then this particular whole group attached here, okay, including the C alpha. So this beta carbon is also asymmetric as well. Okay, so it has actually if we talk about the amino acid which has two chiral centers or two asymmetric carbons, then one is isoleucine which has two asymmetric carbons here. And similarly, you also have threonine here. If you see, this is C alpha, the one attached to NH3 plus and CO minus, the regular amino acid backbone. However, the C beta one here is attached to one H atom, one OH group, one CH3 group, and this complete thing here. So eventually this also has, this is the beta carbon. So threonine also has two asymmetric carbon or two chiral centers. Okay. Yeah, this is what I was showing here, what I told earlier. And yeah, so for all these longer chains, longer side chains, usually this is the alpha carbon. So how you name is eventually the next carbon attached to it is beta, then gamma, then delta, then epsilon. Okay. So for lysine, you go all the way till epsilon, right? I'm really sorry. Please bear with me wherever I'm just slowly progressing and then seeing wherever I have to explain the things, I will explain it. Yeah. So, and all these 20 amino acids have actually short forms and one letter codes as well. Okay. So, if glycine, like there are three lettered short forms as well as one letter code for each of the amino acids. So, for glycine, we have GLY or G as one letter code. For alanine, we have ALA or A, capital A as one letter code. For proline, we have PRO or P as one letter code. For valine, we have V or VAL. Then you have methionine, which is MET or capital M. Isoleucine is ILE or capital I. Leucine is L or like LEU or capital L. Then if we come to phenylalanine, PHE or capital F, tyrosine, TYR or capital Y, tryptophan, TRP or W. Then you have lysine, which is LYS, the one letter code is K, arginine, ARG, one letter code is capital R, histidine, HIS or capital H, and then you have serine, SER or capital S, threonine, capital T, THR, cysteine, CYS or capital C, asparagine, ASN and capital N, glutamine, GLN or capital Q, and finally these two acidic amino acids, like negatively charged amino acids, aspartate or aspartic acid, represented as capital D or ASP, and then glutamate or glutamic acid, GLU or capital E. Okay. So yeah, we looked at isoleucine and threonine having asymmetric beta carbon centers or chiral centers, and in general, so the residues also the aspartate and glutamate residues which we looked at earlier. So these are actually the aspartate and glutamate are the amide forms. Okay, this asparagine and glutamine are the amide forms of aspartate and glutamate. If we go back a little and look at the structure, so this is asparagine, this is glutamine, and this is aspartate, and this is glutamate. So what is happening here is in aspartate you have CH2COOH. Okay. Now OH goes off and NH2 comes in. So asparagine is actually the amide form of aspartic acid. Similarly, in case of glutamic acid, you have CH2, CH2, then COOH. So here the OH goes off. So C double bond O and then NH2 comes in. So glutamine is actually the amide form of glutamate. Oh, all right. And yeah, so I was talking about proteins, the aromatic amino acid residues absorbing at 280 nanometers wavelength. You can see that these tryptophan, tyrosine, and phenylalanine, phenylalanine actually out of these three aromatic residues, these three are the ones which show absorbance at 280 nanometers, and we can actually measure the content or amount of protein in a sample. Okay. However, out of all these three, tryptophan gives the highest absorption. So, in case if any protein has tryptophan residues, greater number of tryptophan residues, it will have and it is present in good amount of content, like in a quite a good amount, quite a good concentration in the sample. Then eventually you will have a higher OD value or absorbance, and tryptophan gives the highest absorbance, followed by tyrosine, and phenylalanine's characteristic is a little like it's much lower, like out of all these three aromatic amino acids, phenylalanine absorbs the least. However, it still is better than the other polar or other amino acid residues. Okay, it can show absorbance at 280 nanometers, but the highest is shown by tryptophan residue. Right. So in general, when we talked about nucleic acid concentration, we saw that nucleic acid shows absorbance. The all the residues, pyrimidine and purine residues, show absorbance at 260 nanometers. Now, in general, whenever you purify, suppose based on the experimental requirements, suppose I need to purify nucleic acids or I need to purify proteins, all right. So now I need pure nucleic acids from the sample. Suppose we talked about plasmid DNA, right? So for plasmid DNA thing, I need pure plasmid DNA. So for that, sometimes what also happens is, because we need pure samples, we check the like absorbance at some other ratios, like other wavelength, and take the ratio to eventually check for the contamination by other types of biomolecules or macromolecules or some other solvents. So similarly, because proteins absorb at 280 nanometers and nucleic acids absorb at 260 nanometers. So eventually we take this ratio also to check the purity of the purified nucleic acid. Okay. So what happens is, suppose you see that the overall ratio should actually range between 1.8 to 2. Okay, for this particular ratio, A260 by A280 ratio should actually range from 1.8 to 2. However, what happens is, in case there's protein contamination or like huge protein contamination, so this denominator will be greater definitely, and hence you will have the net um, ratio will lower down, and this will kind of indicate that the nucleic acid, purified nucleic acid, is really having some contamination, some protein contamination as well. So because we are talking about ratios here, so there's another ratio which is A absorbance at 260 nanometers divided by absorbance at 230 nanometers. So at 230 nanometers, usually we check the contamination by some organic solvents or polysaccharides. Okay. Um, all right. So, because we are talking about absorbance in the previous thing also, like nucleic acid and all these things, we usually use spectrophotometer to measure the absorbance under UV light. So, what in general happens is, and this overall measuring the absorbance thing, it is based on Beer-Lambert law. All right. So what happens here is, suppose if we talk about the setup inside a spectrophotometer, so there's a UV lamp, okay, which gives off light, and then there's a monochromator, which eventually lets only a single wavelength of light to pass through it. And when we talk about that particular single wavelength of light, if we talk about that, if we represent the intensity of that particular incident light as I naught, then eventually this is a cuvette here, this particular cuboid shaped thing that you see. This is a cuvette here wherein you put your sample, suppose your nucleic acid sample or your protein sample inside, which is having a particular length. Okay. So this is sample cuvette having a certain concentration of your macromolecule or like protein or nucleic acid of your interest, where this particular dimension, which is L or D, sometimes it is also measured like represented as D, which is the path length, usually in centimeters. Okay. So eventually, whatever sample is inside, it will absorb some of the incident light and transmit the rest of it. So whatever the rest of the light is transmitted, that is that if we represent it as I, as the intensity of the transmitted light, then eventually that amount of transmitted light is detected by a detector. Okay. So it has been found that this particular absorbance is actually directly proportional to the concentration C here, concentration of the molecule of interest or concentration of the sample here. Okay. As well as it is directly proportional to the length or path length of the cuvette here. Okay. And when we the proportionality constant here comes up, and then eventually you have molar absorption coefficient. Okay. And transmittance, if we talk about the transmittance, how much light has been transmitted, then it becomes intensity of transmitted light I divided by I naught, which is the intensity of the incident light. Okay. So this particular transmittance, the log of this particular thing, if we talk about the reverse of this ratio, that is absorbance, and log of this ratio is directly proportional to CL, and this absorbance eventually when we bring the proportionality constant, that is, this is the molar absorption coefficient. Now this is a characteristic of every, suppose if I am talking about insulin, okay, so then this will be characteristic for, this will have a characteristic value for insulin, for hemoglobin, or whichever protein in question we are talking about, or in general nucleic acid, etc. Okay. And when we talk about the, and it is called molar absorption coefficient. And if we talk about the, so absorbance in general, or transmittance or absorbance in general, doesn't have any unit, because it is basically the same quantities, same units gets cancelled out, right? However, when we talk about the units of molar absorption coefficient, then you have, if we talk about E epsilon here, will be equal to A upon Cl, right? Cl moves here. And so what will eventually come up is, concentration is usually measured in molarity, okay? Like moles per liter. So it will happen, either you measure, write it as molarity inverse and path length inverse, which is in centimeters, or if you break molarity, which is number of moles per liter, then it becomes liter, because it is moles per liter, right? And then it will get switched off, as in switched in this case, because it is coming off as denominator, so liter goes up and moles goes down, right? So eventually you will have the units for molar absorption coefficient as liter per mole per centimeter, okay, or per path length, whatever is the, usually it's in centimeters. So you have to also see sometimes things get represented in, sometimes for any kind of problem solving question, sometimes they give it in meters and stuff. So make sure you are taking care of the units. Okay. Yeah. Mhm. All right, it's getting messy. Maybe I will stop here and see. Okay, so every amino acid, if we talk about, so it has acidic and basic properties, and it will have characteristic titration curves. Okay. Now, this is, this will show basic property, this will show acidic property, right? And so this will have its own pKa value, this particular group will have its own pKa value. Now depending on what type of R group the amino acid has, it will eventually have, either suppose you have asparagine, lysine, etc., all these stuffs are there, and suppose you have aspartic acid or glutamic acid, so eventually the R group here will also add into the net charge on the amino acid, right? So, but at least you will have, for an amino acid, two pKa values. Suppose we label it as pKa1 and pKa2. So there are two acid dissociation constant values. You will be having at least two values, right? So and this particular charge, if we talk about the overall charge on an amino acid, and what, whether it will exist as NH2, or whether it will exist as NH3 plus, whether this will exist as COO minus or COOH, it also is dictated by the pH of the surrounding medium where these amino acids or amino acid sequence is existing. Okay. So what happens here is, eventually if, now suppose if we talk about, um, this particular amino acid here, where we also, one more thing is there, like depending on the pH of the medium. Now, whether if there are more H plus ions in the medium, then definitely this will exist as, it will donate its lone pair and eventually exist as NH3 plus. And if there are more H plus ions in the medium, NH3 plus, and it will exist as COH form here. However, if the H plus ions like or OH minus concentration increases, then eventually what will happen is OH minus, so this will tend to give away its H plus ion, this carboxylate group, and it will exist as COO minus form. Okay. However, this, because there's less amount of H plus ions in the medium, so this will exist as NH2. So overall, if you see, the COOH eventually exists as COO minus in the medium. So now suppose we talk about this particular thing where at a particular pH, this particular amino acid with R group here, CH, NH3 plus and CO minus. So at this particular pH, NH2 takes up one H, like as in the lone pair on N is being donated to one H plus, and eventually it exists as NH3 plus, and COOH gives off its H plus and exists as COO minus here. So the net charge, if we talk about, is 0 on this amino acid. Now suppose, now if the pH is 1 in this case, okay, and if the pH is 1, so then what happens is eventually the surrounding medium becomes more acidic. There's more H plus ion in the surrounding. So what happens is eventually this COO minus takes up that H plus ion from the surrounding and exists as COOH. However, this still exists as NH3 plus. So the overall charge on this particular amino acid is +1. Okay. And similarly, if the pH increases, suppose it goes like increases than this particular value, which is pH 6 here, then what you have is it is becoming like less acidic, and so we will expect lesser amount of H plus ions in the medium. And hence what will happen is, because there's lesser amount of H plus ion in the medium, the COH group will donate its H plus ion, and NH3 plus will also, the H plus from this NH3 plus here will be given off to the medium, and overall the net charge on this amino acid will be -1. Okay. Now, when we talked about the previous example where we looked at pH 6, where it is existing as a neutrally charged molecule, right? So in that case, whenever on an amino acid, whenever the overall charge is 0, it is called a Zwitter ion. Okay. And the pH at which any amino acid exists as a neutrally charged molecule, or it has an overall charge of 0, then that particular pH is called pi, or isoelectric point of that, for that particular amino acid. Okay. So for the previous example that we looked at pH 6, it was existing as, that was just an example. So at pH 6, it was existing as a neutrally charged molecule. So 6, for that particular amino acid, the pi will be 6, because that is the pH at which it is existing as a Zwitter ion. Basically at this now, amino acids, due to their charged properties, they tend to, so at this particular pH, at this state, in this Zwitter ionic state, if it has to move across an electric field, it won't be moving beyond that particular point. Okay. So it, because it is neutrally charged, it won't be either moving towards the negatively charged electrode or positively charged electrode. It will stay at that point wherever it has migrated till that particular point. Okay. However, if it was, only positively charged, then it would try to move towards the negatively charged electrode. If it was negatively charged, then it would try to move towards the positively charged electrode. However, when it is having like whenever it reaches the isoelectric point or exists as a neutrally charged molecule, then it, like in an electric field, it won't be moving anywhere. It will stay at that particular point wherever it is present. In a, suppose we are separating it on an on a matrix or somewhere, then in that case it won't be moving to any of the electrodes. Okay. This is what is being talked about, like net negative charge and net positive charge actually depends on the pH of the medium where these amino acid sequences or amino acid residues are existing. Okay. Now here, the same thing that we looked at for that example of amino acid earlier. Now for glycine, so due to these charged properties, each amino acid actually has characteristic titration curves. Okay. So this is the pi value where it exists as a neutrally charged molecule, the glycine residue. And then there are again, when you really lower the pH or kind of increase the pH, then the overall charged state of the amino acid in question changes. Okay. Now suppose this is the neutrally charged glycine residue, right? Now what happens is when you uh, kind of move towards this side where OH minus concentration decreases, that means the medium is becoming acidic. So more H plus is present in the surrounding. So what happens in that case is this CO minus accepts H plus ions which are actually abundant in the medium now, as well as this lone pair on nitrogen is being donated to taken up by another H plus ion, and eventually the overall charge, net charge on this glycine molecule is +1. Okay. However, if you see towards this side, so here it exists as a positively charged residue. Glycine residue in this particular region exists as a positively charged residue. Okay. And if we move towards higher OH minus concentration, what eventually happens is as OH minus concentration increases, H plus ion concentration reduces. So this NH3 plus gives off the H plus ion, thereby existing as NH2, and COO minus exists like COH gives off its H plus ion and exists as CO minus, and eventually the net charge on this particular in this particular state is -1 for the glycine molecule at higher pH. Okay, so in this particular region it is existing as a negatively charged molecule. Okay. So in general, what happens is now because it has, we are talking about glycine, which is the simplest amino acid. So it has two ionizable groups. So for two ionizable groups, it will have two acid dissociation constants, right? So for that, what happens is eventually this was found to be uh, to get the isoelectric point of this particular glycine amino acid where it exists as a neutrally charged molecule. How you can calculate this is pK1, which is this particular acid dissociation constant in this particular pH, plus pK2, which is the dissociation constant in this particular pH, which is and take the average of that. Okay, so in this case, in this example, it is 2.34 plus 9.60 divided by 2, which gives off 5.97, which is the isoelectric point, which is the pH at which glycine molecule, glycine amino acid exists as a Zwitter ion or a neutrally charged molecule. Okay. Now the same thing here that we were talking about earlier. So here, if the pi eventually is greater than, like the pH is less than the pi of value, like isoelectric point of the amino acid, then if pH is less, then it will have more H plus in the medium and it will exist as NH3 plus and COH. So the positively amino acid will be positively charged. If the pH is more than the pi value, then it will tend to give away the H plus ions, and eventually the amino acid will be negatively charged. Okay. Now in general, when we talk about this, if we look at this particular graph, what is happening here is, wait, So in general, when we look at the slope of, in general, when we talk about the slope of any particular graph, this is y by x, right? This is also a graph here, this titration curve that is being shown. So now suppose there are three samples, okay? So it is having a certain angle here, and for that slope thing, what we have is tan theta, or in general, slope is represented by changes in the perpendicular axis divided by changes in the horizontal axis, right? So delta y by delta x. So if the greater the slope, I mean if the slope is greater, then it is actually very steeper, right? So eventually, see here, theta a here in this case for the A sample, you have a greater slope and steeper slope, right? So what will happen in that scenario is any small change, when the slope is really steep, if you see this particular region here, the pi region, if we talk about this particular pi region here, so any small change in the delta x value will lead to a very large change in the y axis, right? Now if you see, um, here, suppose this is at one concentration of OH, when it is 1 here, you see it is, suppose, this particular value. However, if you even increase it to 1.2, still it moves to much large, it gives off a much larger change in the y-axis, right? So even for a very small change in the medium, surrounding medium, it will give a very large change in the uh, like y-axis here, okay? So because the slope is too steep here, okay. However, if you look at this particular regions, these blue colored regions here, what is happening here is the slope is very less steep, right? As compared to this one, you see the slope is really less. So you see that for a greater number of, like even if there's a large change in the x value, x region here, still it still maintains the pH at the same region almost, right? It's the exact opposite to what was seen in this particular region. So these are actually the buffering regions for that particular amino acid. Okay, we talk about buffers and stuff, right? So what is actually a buffer? Buffer is any kind of solution which maintains the pH at a specified range, in a given range, right? So this is the range where for this particular glycine amino acid, it shows buffering capacity. Okay, because for even large changes in the OH minus concentration, it still gives, like it still maintains the pH at an optimum range, right? In this particular case as well as in this particular region as well. Okay. So this is what you actually get the idea from titration curves. Um, Yeah. So that is what I was talking about here. Like this is a very steep area, very steep gradient, and this particular region is very sensitive to pH. Even a slight change in the concentration of OH minus ions will lead to very large changes or drastic changes in the pH. Okay. Yeah, the same thing which I talked about. These are the buffering regions where even for larger changes in the concentration of OH minus, the pH more or less remains the same. All right. However, that was for glycine residue, which we looked at earlier. But here in this case, now, so now when it becomes a little more complex, when the R group has some other residue attached to it. So here you see you have an acidic region here for glutamic acid and for histidine. So it will again have an additional, so pK1, pK2 are there definitely for the COH and NH2 groups attached to the C alpha carbon, as well as because of the presence of carboxylate group or the positively charged, these, in case of histidine you have this particular N here. So all these will eventually add another, will give another acid dissociation constant values, and eventually the titration curves will be more complicated and more complex, but you will be able to find out the buffering regions in these from these graphs as well. And so that is how we eventually find out the pi values and buffering regions for different amino acids by, like, from their titration curves. Okay. And also you have, so we talked about the 20 naturally occurring amino acids, right? And but however, there are certain uncommon amino acids which also exist in nature, which are actually modified or modified forms or derived versions of these 20 naturally occurring amino acids that we talked about. So one is 4-hydroxyproline. So proline is already there. Just in case of like only there is an addition of this OH group here. So mostly this particular amino acid, this uncommon amino acid 4-hydroxyproline, is present in plant cell walls, in collagen, connective tissue matrix. Then you also have 5-hydroxyl lysine. So again, so lysine residue, uh, just here, this is the C alpha carbon attached to H, CO minus and NH3 or NH2. And then you have, this is the beta, gamma, alpha, beta, gamma, delta and epsilon, and you have finally NH3 plus. So lysine is a, this is in general the backbone of lysine. However, in case of CH2 here, one H is replaced by OH, thereby forming 5-hydroxylysine, which is an uncommon amino acid but derived from the positively charged lysine residue, and this is found in collagen matrix and all. Okay, then you again have 6N methyl lysine. Same here, the lysine residue is modified at the NH3 plus, terminal NH3 plus here, and you have, this is actually found in contractile proteins like myosin. Then you have gamma carboxyglutamate. So the glutamate residue is actually modified by where another carboxylate group gets attached. So this is found in mostly calcium binding proteins or prothrombin, which is a blood clotting factor. Then you also have selenocysteine. Cysteine residue here, like in general for a cysteine residue, what you have is sulfur attached. There's a sulfhydryl group S and H here. Okay. But however, in place of S, a selenium atom comes in the picture and eventually thereby forming selenocysteine. And there's another a little heavier uncommon amino acid, which is desmosine, where 4 lysine groups are present, okay, and attached to this particular ring here. And these are actually, this particular amino acid is found in fibrous proteins like elastin. Okay. Next, we will talk about peptide bond. So, and again, depending on how many amino acid residues are attached, it can, 2 amino acids, it can be dipeptide, tripeptide for 3 amino acids, and eventually if it's a longer chain, it will be a polypeptide. Okay. And the sequence eventually gives off a structure, and structure actually dictates function for a protein. Okay. Now for a peptide bond, what is happening here is you have one amino acid residue with R group attached to it and another amino acid residue with another R group attached to it. So here COOH from the previous carboxyl group of the amino acid and the H from the next amino acid residue gives off, there's a dehydration reaction or condensation reaction takes place, H2O is given off. Eventually what you have here is this C alpha carboxyl group attached to double bonded O, and then finally this N from the other amino acid. This particular bond here, CONH bond, this particular thing here is a peptide bond. Okay, which is formed as a result of condensation reaction and loss of H2O molecule. Okay. And so peptide bond. So in this case, this is a dipeptide because there are 2 amino acids involved. And what else? So yeah, if we talk about polypeptide, then you have a series of different amino acids with different kinds of side chains. And however, what you also see is eventually the N terminal has NH2 group and in one side and C terminal has COH group at the end. How much ever bigger the polypeptide is, even for this dipeptide, you see at one end there is NH2 group which is called the N terminal end, and in the on the other end you have like the extreme two ends. We are talking about the last parts of this particular chain. So another end is having COOH group where it is called a C terminal end. Okay. So this is what I think. Yeah. This is N terminal end of this polypeptide. Similarly, you have C terminal end of this polypeptide here. Okay. Anything else? Yeah. So now in general, um, if we talk about a peptide bond has a partial double bond character. Now, when we talk about here, CN is a single bond or sigma bond. However, C double bond N has pi bond characteristics, right? Now, if you look at this particular figure here, this particular N here, um, it may be here, you can see that it also has a lone pair over here. Now, what will also happen is this will come here and this particular cloud electron will eventually get delocalized. Hence, this N will also have a partial delta plus charge, and this oxygen over here eventually gets a delta minus charge. So the bond gets delocalized, double bond bonded character gets delocalized. So these are the resonance structures. Okay, this one and this one, and eventually this delocalization of charge leads to partial double bonded character for this peptide bond here. So this is the peptide bond, C double bond O of one amino acid and NH from the other amino acid, eventually condensed via dehydration reaction. Okay, so how did they actually come up with this thing that it might be having a double bonded, like partial double bonded and resonance character? Because they saw that Pauling and Corey, they saw that CN peptide bond, this particular single bond, sigma bond, is actually shorter than CN bonds in simple amines. Okay. However, atoms that are part of the peptide group of a bond are coplanar. So these were actually X-ray data, okay, which indicated a resonance between the carbonyl oxygen and amide nitrogen. Okay. So in general, single bond is much larger, and if it is having a full double bonded character, pi bond character, then it will be much shorter. However, the characteristics of this particular area showed it didn't show complete single bonded lengths or double bonded lengths as well. It showed something intermediate in between. So that's why these were all experimental X-ray data. So that's why that the data indicated a resonance between this like carbonyl oxygen and amide nitrogen. This is a double bonded character here. Okay. Something else. Yeah, so this is what I was showing, like the lone pair is given off here, then this goes off here, so there is delocalization of the uh, electrons over this region. Okay, so if this is given off here, this is taken off there, then eventually O minus and N plus. So there is partial double, like partial charge, like positive charge, partial charge, delta plus on N atom and delta minus on oxygen atom here. Okay. And also, if you see these four atoms, so this actually, the X-ray data also showed that these four are actually coplanar. They exist on a single plane. Now, if we talk about the C alpha of one amino acid and C alpha of the other amino acid here, these can actually move out of the plane or go elsewhere into the plane or out of the plane. However, these four atoms here, C, O, and H, the ones participating in peptide bond formation, are actually coplanar. They exist in a single plane. Okay. Mm, yeah. So that is what we were talking about. The peptide bond length is only 1.33 angstrom, which is actually shorter than the usual CN bond length of a longer distance, 1.45 angstrom. However, it is longer than the value of 1.25 angstrom, which is average for C double bonded N. So this value lied in between, lay in between these two single bonded distance bond length as well as double bond length. So the peptide bond appears to have approximately 40% double bonded character. Okay. So that is where they came up with the idea that it is getting delocalized. Sorry. And what else am I missing? Yeah. So in general, when we talk about the trans and cis forms of, now here, in case of, so this is the peptide bond here, CONH, okay? And so what happens is this C is attached to this C alpha here, this N is attached to C alpha of another amino acid, right? So this is, suppose this is attached to amino acid 1 and this N here is attached to C alpha of amino acid 2. Now these C alpha and the eventually the atoms or groups attached to this particular C alpha of amino acid 1 and similarly for amino acid 2, these are really bulky groups. Okay. Now in general, you know that if these two were to exist at the same side, then there will be repulsion between the bulky groups. So thermodynamically or energetically, these are more stable in the trans form. Okay, trans form, like these are present in the opposite areas. So there's lesser steric hindrance or lesser steric interactions between these two bulky groups of two different amino acids. Okay. So as compared to the cis form, where you will have comparatively greater amount of steric hindrance. Just imagine C alpha. It's not only C alpha. There are R groups attached to it, bulky groups attached to it, right? So on the same side of the double, like peptide bond, you will have more amount of steric repulsion. However, when we talk about proline, if you remember, proline had a heterocyclic chain here. It didn't have free NH2 and free COH groups like this. So the NH was actually inside a heterocyclic chain. So in that scenario, eventually because it doesn't have free groups as such, so the intrinsic stability and energetically stable form will be like it is comparable. We can't really say for proline that the trans form is more stable than the cis form. The intrinsic stability of cis and trans isomers for prolines after a peptide bond will be comparable. Okay, because it has a cyclic side chain. So cyclic side chains actually diminish interatomic repulsion. Okay, I guess this is the end. Yeah. Now, now these are the normal slides. We will go through it, and the video is over. So in general, how do you really check for the presence of amino acids? Okay, what are like different amino acids have different characteristic properties. We looked at the side chains which eventually give them that property. So there is one method which is chromatography. So chromatography is one of the most useful techniques available for separating closely related compounds in a sample, in a mixture. So here the separation is actually affected by the differences in the equilibrium distribution of the components between two immiscible phases. One is the stationary matrix and the mobile phase or the solvent, which moves up eventually along the matrix. So the differences, so whatever is the sample, like there are suppose A, B, C components in a sample. So one solvent is moving up across the matrix. So the A might be more soluble with the solvent. So eventually it will try to move up quickly as compared to C, which is very less interactive or very less miscible with the solvent. Okay. So these differences in the equilibrium distribution are a result of nature and degree of interaction of the components with these two phases. In general, the matrix or the stationary phase is a porous medium like silica or alumina through which the sample mixture percolates under the influence of a moving solvent or the mobile phase. There are a number of interactions between the sample and the stationary phase, and these have been well exploited to effect the separation of compounds. Now, thin layer chromatography, what happens in here is um, it actually provides qualitative information with careful attention to details, and it is possible to obtain quantitative data. So TLC is a technique which separates and identifies compounds of interest in a mixture. So again, TLC plate, if this is the TLC plate which is coated with a matrix, which is the stationary phase in this case is either silica, mostly silica gel, which is kind of stuck on. The silica gel is actually layered on a glass or aluminium for support, and this gel acts as a stationary phase and the solvent mixture acts as the mobile phase. So in an ideal solvent system, the compounds of interest are soluble at to different degrees, and hence this results in different mobilities of each component in that mixture and eventually leads to different partition coefficients. Okay. And so what happens is you spot your mixture here, and it has suppose these four different compounds, and you dip this TLC plate in a solvent, like inside a container you dip that inside a solvent will start moving up, and eventually, so if you see this particular brown spot at the topmost part, so then this is more soluble with the solvent as compared to the yellow one, which is less soluble with the solvent and hence moves up the least as compared to all these four here. Okay. And so this particular migration from the line of application, line of spotting, you can actually measure the different RF, like these values, where the distance moved by the substance from the origin divided by distance moved by solvent from the origin. This will eventually give you an idea of how far the mixtures are moving, and each will have a characteristic distance that it travels. Okay. And eventually you can uh, kind of deduce from there that okay, this is the component present in the mixture. Okay. So in the simplest form of the technique, a narrow zone or spot of the sample mixture to be separated is applied near one end of the TLC and allowed to dry. Then you dip it in the solvent mixture, and as the solvent moves up towards the end of the strip, the mixture that you have spotted also separates eventually. So this is called as the development of TLC plates. And how is the separation dependent? It is dependent on several factors like solubility. The more soluble a compound is in a solvent, the faster it will move up the plate. In this case, the brown one is more soluble as compared to the other components. The attractions between the compound and the silica, the more the compound interacts with silica, the lesser it moves. So this yellow one interacts more with the silica, which is the stationary matrix here. So it stays here and eventually moves the least. The size of the compound, also the larger the compound, the slower it will move up along the plate. Okay. And the plate is removed after an optimal developmental time and dried, and the spots and zones are detected using a suitable location reagent. Okay. So now for the common 20 amino acids, so these are all alpha amino acids because they have carboxyl group and an amino group bound to the same carbon, alpha carbon atom. They differ from each other in their side chains or R groups, which vary in structure, size, and electric charge. So the interaction of the amino acids with the stationary phase like silica can vary depending on their R groups. The amino acid that interacts strongly with silica will be carried by the solvent to a small distance, whereas the one with less interaction will be moved further. So by running the compounds alongside, now if you also have a reference compound that you know that it will run up to this part and it definitely has glycine in it, and the other mixture test sample, you don't know if glycine is present or not, depending on how far this particular component gives off the signal, you will eventually understand that if your test sample has glycine present in it or not, just as an example. Okay. Now, since amino acids are colorless compounds, ninhydrin solution is there, which is used to detect the presence of it. So the TLC plate is sprayed with ninhydrin reagent and dried at a higher temperature for some time. And what this does is this reagent reacts with alpha amino acids that results in purple colored spots. Okay. Now, if we talk about the structures of amino acids, the general structure of amino acid, which talked about the sequence, regular like primary sequence, one after the other, the amino acids are linked by the peptide bonds, okay, thereby forming a polypeptide. If we talk about this, this is the polypeptide sequence, amino acid sequence. Simple amino acid sequence is the primary structure. Now, these primary structures, because they have different kinds of R groups all around, right? And depending on, they have planar peptide bonds in between, and the other groups that are attached to the peptide bonds, I mean, attached to the atoms participating in peptide bond formation, those are mobile. Those can actually move here and there based on what are the steric residues, I mean bulky groups present in each amino acid. So depending on that, it can take up different forms: secondary structure, tertiary structure, and quaternary structure. Okay, eventually affecting the function of a protein. So secondary structure, what happens? The amino acid interacts with each, like with each other, and as a result, peptide chain folds into a secondary structure. So these secondary structures are the building blocks for tertiary structure. So if we talk about secondary structure, it can actually uh, be majorly divided into three types, like alpha helices, beta sheets or beta strands, and turns. Okay. Eventually, turns are the kind of intermediate portions in between these helices and sheets or strands, which helps in changing the directions of these strands or helices. So these secondary structure, if we talk about turns, they have no definite structure, and these are present in protein structure to change the direction of the running polypeptide. These are also found in places where we have to connect the successive alpha helices and beta sheets. The number of amino acids and their preference in turn is not consistent. Two proteins can adopt similar 3D conformation by changing the length and keeping the amino acids in the turned region of the structure. Okay. Similarly, if we talk about the tertiary structure, so these tertiary secondary ones, these are the beta strands, these are the helices. Okay. So these eventually again fold among themselves to give rise to a tertiary structure. Okay, which is much more organized, and eventually each of these particular organized ordered regions come together to give rise to a quaternary structure, which are actually multiple polypeptides constituting a protein, and they actually come together to form this quaternary structure. Okay, if we talk about hemoglobin, suppose it has two alpha chains and two beta chains. It is also tetrameric in nature. um, so that is the two alpha chains and two beta chains coming together in the coming together to form the functional hemoglobin structure, that is the quaternary structure. Okay, so now proteins are actually polymers of amino acids, and we looked at the peptide bonds and stuff. But bond between NC alpha and CC alpha are able to rotate through dihedral angles designated by phi and psi. Okay. Now, if we talk about phi and psi angles here, so this is uh, this is one amino acid. This is another amino acid. This is another amino acid. Okay. This one is this N. This C alpha is from amino acid 3, suppose. This C alpha is amino acid 2, okay, attached to N and CO here, okay. And again, this particular one is having, like, suppose amino acid 1. Now, what is happening here is this is a peptide bond uh, here, and this is another peptide bond here, uh, where these CONH are present in the same plane. This is the peptide plane, amide plane here. And however, the regions here, these are easily these, so any rotation around the double bond or partial double bond character is not possible. However, the single bonded regions here can be easily rotated. So this NC alpha here is the phi bond, and CC alpha is the psi bond here. Okay, so eventually G.N. Ramachandran, he actually gave this Ramachandran plot, which is kind of a dictionary for structural biologists, especially studying protein structures. So here you can actually, based on his helices and stuff, he actually plotted this where you can see the regions of these psi and phi angles, these phi angles and psi angles, where you can actually see what are the permissible regions where these particular different kinds of secondary structures can exist, and which are the non-permissible regions. Here you don't see, for all these values of psi and phi angles in these regions, you won't be able to see a viable structure existing, protein structure existing. Okay. Sorry. And another thing to note is when we talk about orthologs and paralogs. Okay. So, in case of orthologous genes or orthologous proteins, orthologous genes are the homologs in different species which eventually got separated by speciation, and paralogs are the homologous genes found within a single species but eventually got kind of little varied due to duplication. So orthologs carry out the same function as the ancestral gene from where they diverged, and paralogs can have different functions other than the function of the ancestral gene. So orthologous genes are a result of speciation. Paralogous genes are due to genetic duplication event. So orthologs are present in different species. Paralogs are present within the same genome of a single species. Okay. And orthologs are similar to the ancestral genes. Paralogs are combinations of ancestral and new genes. Now, suppose this is the very early ancestral gene in question. Gene duplication happened, and alpha chain, there is a gene encoding alpha chain, there is a gene encoding beta chain of a certain protein. Now, in different species, you have this alpha chain in frogs, in chicken, in mouse. So, all these alpha chains in frog, chicken, and mouse are orthologs of each other. Okay. However, when from the same gene, alpha chain and beta chain emerged, right? And then this beta chain again is existing in mouse, chicken, and frog. So this beta chain in each of these organism, different species, are orthologs of each other. However, because the alpha chain and beta chain originated from the same ancestral gene, and eventually, you know, due to gene duplication and stuff, they became different. And here, then what will happen is the alpha chain in this particular mouse and beta chain in this particular mouse are paralogs. If we trace back the ancestry and evolutionary history, we'll find that they actually had originated from the same ancestral gene. Okay. So similarly, if it's orthologous and paralogous genes, similarly you have protein orthologs and protein paralogs as well. Okay. So, as a general rule, orthologous proteins tend to perform the same function in different species, while paralogous proteins tend to have diversified somewhat in function. Duplication is a very common way in which evolution gives proteins the freedom to develop new functions. Now, for example, chymotrypsin serine proteases are orthologous to each other. Chymotrypsin is an enzyme, serine proteases are also an enzyme, and these are orthologs of each other. They retain not only the same general function, which is proteolysis, and they have a way of, kind of digesting a peptide bond, but also the same substrate specificity. It cleaves at positions following aromatic side chains. Chymotrypsins are paralogous to trypsins, however, and elastases, which are again two different enzymes. So these proteins share the same general serine protease function, but they have evolved different substrate specificities. So these proteins also have paralogs which have lost all protease activity. Okay. Now, if we talk about the function of a protein, determining the 3D structure of protein is important for understanding its functions. So, in prokaryotes and eukaryotes, protein is the main molecule to perform many different functions like enzymes to catalyze different chemical reactions, adapter molecule for different ligands, messenger molecule to relay the signal within the cell to produce factors to generate defense response against pathogens. So proteins can function as enzymes or biological catalysts. It shows reversible binding of protein to ligands, metabolic intermediates, or as substrates. It can act as cell surface receptors. We have been talking about receptors like glucose receptors as well in the previous slides, previous session. Phagocytosis, antigen presentation, antimicrobial killing, inflammatory responses, tissue repair and modeling. Everywhere, proteins are really, really important for for performing all these functions. Okay. Now, in order to uh, determine the overall size or uh, kind of molecular weight of a protein, there are different kinds of techniques. Like one is SDS page, like we looked at agarose gel electrophoresis to look at nucleic acids or look at the integrity of plasmids after isolation, after purification. Similarly, you can run SDS page, which is a denaturing polyacrylamide gel electrophoresis, where it actually separates proteins according to their molecular weight based on their differential rates of migration through a porous matrix under the influence of an applied electric field. So in general, when we talk about the movement of any charged species through electric field, it is determined by the net charge, molecular radius, and magnitude of the applied field. So, but however, proteins are folded. So the problem is their net charge or the molecular radius is molecular weight dependent, right? And the net charge is given or decided by the amino acid composition, which is eventually the sum of positively and negatively charged amino acids, or not necessarily positively or negatively charged amino acid. Eventually the hydrophobic residues or polar or non-polar residues can also help in uh, protein folding, eventually leading to masking of some positively charged or negatively charged residues as well. Okay, and it also decides the molecular radius to eventually which decides the protein's tertiary structure or the overall hydrodynamic radius. So in native state, different proteins with the same molecular weight would migrate at different speeds in an electric field depending on their charge and 3D shape. To separate proteins in an electric field based on their molecular weight only, it actually destroys the tertiary, either you can destroy the tertiary structure by reducing the protein to a linear molecule, then only you will have an unbiased way of separating the separating the different protein fragments. Or what you do is you mask the intrinsic net charge of the protein by coating it with a uniformly charged molecule. So that is what SDS page does. What happens is SDS is sodium dodecyl sulfate, which is a detergent present in the buffer. And also while preparing the samples are boiled. So high temperature can also denature proteins. And a reducing agent like dithiotretol or beta-mercaptoethanol is used to break down disulfide bonds. So eventually all these treatments disrupts the tertiary structure of the proteins, and the folded proteins are down to are linearized. Okay, and SDS eventually coats the protein with a uniform negative charge and masks the intrinsic charges on the R groups. And the dominant factor in determining an SDS coated protein's mobility eventually becomes the hydrodynamic radius or the molecular radius. Okay. Now, in general, some of you might know, or probably almost all of you might know, that for an SDS page, you have a discontinuous buffer system. Also, you have two types of gels here. One is the stacking gel, where you have low concentration of acrylamide and where the proteins don't really separate here, this area. And then finally, you have the resolving area or running gel, which has higher acrylamide concentration capable of retarding protein movement. Now, stacking gel, so we looked at in the earlier ones, looked at the titration curves for glycine, right? And how it exists in different charged forms. So that particular thing is again used here, applied here to run to separate out different kinds of proteins. Okay. So the stacking gel has pH 6.8. So in this case, pH 6.8, we looked at the PI value of glycine as 5.97, right? So when pH is 6.8, so glycine becomes predominantly its zwitterionic form, or like it has zero charge. And so glycine actually moves very slowly in the electric field, or it's not moving at all, kind of. Okay. However, you have chloride ions from the tris HCl buffer, which actually moves much more quickly towards the positively charged electrode here. Okay, and hence chloride ions form a like ionic front, and which migrates ahead of glycine. And separation of Cl minus from tris counter ion. So tris Cl is there. So tris moves towards the anode, this part here, and Cl moves towards the um, this positively charged uh, electrode here. It creates a narrow zone with a steep voltage gradient that pulls glycine ions along behind it, resulting in two narrow separated fronts, highly mobile Cl- front, followed by the very slow glycine ion, which is in a zwitterionic form, followed by uh, ha- that is there, which is mostly neutral and hence very slow to move in that electric field. So all the proteins in the gel sample have an electrophoretic mobility intermediate between the extreme mobility of glycine and Cl minus. Glycine is too slow and Cl minus is too fast, as well as Cl minus being very small also and lighter comparatively in molecular weight, it will also move much faster. So two fronts sweep through the sample well. So these are the wells here. You see these kind of incomplete rectangles here. So this is where you load your samples, okay, like protein samples. And eventually, these are also called the sample wells, okay. So this suddenly in the resolving area, so all the samples keep on moving through this particular pH, and suddenly in this region, the pH changes to 8.8. Now what happens? pH switches to 8.8, glycine molecules eventually becomes negatively charged, right? Because here it becomes 8.8, so there's lesser amount of H plus ions, and hence COOH exists as COO minus and NH3 plus gives off its H plus, eventually forming NH2. Okay, so the overall net charge is negative on the glycine amino acid. So at 8.8, glycine molecules are mostly negatively charged. It migrates much faster than the proteins. And hence the glycine front now accelerates past the proteins, and proteins are dumped in a very narrow band at the interface of the stacking and running gels. And running gel, we saw that it has increased acrylamide concentration, which actually forms the matrix, and hence the porous matrix slows the protein movement according to their size, and hence resolving or separation of the protein bands eventually starts. So why do we really need the stacking gel? Because eventually the separation and resolving is happening in the running gel only. Because gel wells are nearly, sample wells or gel wells are nearly 1 centimeters deep. So we need to really substantially fill them to get enough protein onto the gel. Now when you are filling, what would happen is sample would sit on top of the running gel as a band of up to 1 centimeters deep. So rather than being lined up together and hitting the running gel together, proteins would all enter the running gel at different times, resulting in very smeared bands. So for runners or for a race, what you want is everyone has different speeds, right? So if one starts running at their own pace, it won't be a good point to judge the race, right? Judge the runners. So all the runners actually have to come at a starting point, and from there the race starts. Okay, that is why you need the stacking gel, so that all the proteins arrive at the running gel at the same time, and proteins of the same molecular weight eventually start migrating as tight bands. Okay, so in the lecture videos we looked at very extensively at ELISA, which is enzyme linked immunosorbent assay, where antibodies were being used to detect any antigen or proteins of interest in the sample. And there were different ways of kind of using these antibodies. Either two different antibodies are being used. However, the overall thing is that these antibodies have specificity against certain regions of the antigen in question, like protein molecule which is present in our sample, and that actually goes and binds, and eventually the signal is getting detected. Either the antibodies are actually bound to some other substrate which is getting cleaved by other enzymes and giving off signals, or similarly this is ELISA here. So similarly proteins in a sample can also be detected by Western blot. So we looked at SDS page, right? So till SDS page you run the gels. Now you transfer this is the SDS page. Okay, these are the separated bands, different bands moving at different molecular weights, right? So also in a matrix, so the really higher molecular weight proteins will actually have more hindrance and more difficulty in moving through the pores, right? So it moves the slowest, whereas the lower molecular weight proteins moves the fastest, moves very easily, so it will have lesser hindrance from the pores from the matrix. So eventually it moves through much faster. Okay. So you will find lower molecular weights protein at the bottom and higher molecular weight proteins on the top. Okay, at the top. Now what happens is you have the separation nicely done. Now what you do is you actually transfer all these bands as such on a particular membrane. Okay, so all these bands eventually get transferred here, and then what you do is against your protein of interest you have antibodies developed. Okay, so those antibodies eventually bind to specific proteins present on your sample, and then same as you looked at ELISA in Professor Vishal Trivedi's lecture videos. Similarly, here also you will have detection from the antibody. Antibodies are either conjugated to some other substrate. Eventually, that substrate is broken down by enzymes, and you will see a chemiluminescent signal, or you have some fluorescent signals. Like different kinds of signals can be there. However, eventually you will be able to, from that signal, you will be able to detect your protein of interest on the, like, on the membrane. Okay, so the immunodetection is same. However, there is an additional thing of membrane transfer after STS page. Okay, so now when we talk about protein sequencing, it enables analysis of amino acid sequences, confirming the protein structure and function, helps in identification of new protein biomarkers and drug targets. It also helps in constructing phylogenetic trees to understand evolutionary relationships. It identifies orthologs and paralogs. It helps in extrapolating the functions to unknown orthologs, facilitating library screening and supporting protein engineering studies. So basically, it examines the covalent structure and amino acid sequence of a polypeptide, of a fully developed polypeptide, which is considered a component of post-translational modifications. And structure of a protein is intricately linked to the sequence of amino acids in that protein. So what are the different methods to sequence a protein? So what you eventually do is many proteins are linked by disulfide bonds. So this disulfide bond is actually broken. Either it actually creates problems in complete sequencing procedure because cleaved amino acids are not released from the peptide chain. So what happens is in the first approach, protein is oxidized with performic acid here, okay, to produce two cysteic acid residues. Okay, and another is reduction by DTT, where again it forms this sulfhydryl residue, and these two, the disulfide bond is broken and H is attached to each sulfur residues, and hence these two can't really get back together to form this disulfide bonds. Okay, now what happens is another approach, protein is reduced by DTT or BME to form 2-cysteine, followed by treatment with iodoacetate to form carboxymethyl cysteine. Okay, so the H is actually given off upon this iodoacetate reaction, and carboxymethylation happens, and this can't eventually get back to this disulfide formation. Okay, eventually this disulfide bond is cleaved. So iodoacetate is commonly used to alkylate free thiol groups during reduction of disulfide bonds. When disulfide bonds are reduced, free thiol groups, this SH bonded like bound SH are generated. Iodoacetate reacts with these thiol groups to form carboxymethylated cysteine residues, effectively preventing the reformation of disulfide bonds. It is actually important in experiments where maintaining the reduced state of cysteine residues is actually necessary for further analysis or protein characterization. Another thing can be simply, cutting or digesting the polypeptide chain. So there are enzymes like proteases and other chemical agents which actually target specific regions of the proteins, like specific amino acid sequences, and they cleave only after a particular amino acid. Okay, like trypsin cleaves after lysine or arginine residues, pepsin cleaves after leucine, phenyl, alanine, tryptophan, or tyrosine residues, like that. And these are enzymes. Now, this is another reagent which is cyanogen bromide, which actually has been found to cleave polypeptide chains after methionine residues. Okay. Now, protein sequencing can be categorized into three main methods: investigating the N terminus or looking at the C terminus, or breaking down polypeptides into shorter peptide fragments. So identifying the N-terminal residue, uh, three-step analysis, where the very first one is derivatization of the terminal amino acid. So this chemical reaction is performed to label terminal amino acid with compounds like Sanger reagent or DFNB. Okay, DFNB, this is the amino acid. So it basically labels the terminal amino residue here, and this gets labeled with DFNB, thereby forming this dinitrophenyl amino acid complex. Step 1. Next comes hydrolysis of the protein. This step here, and so this is the dinitrophenyl amino acid complex. Then hydrolysis happens, where acidic hydrolysis takes place for this dinitrophenyl amino acid complex, and it breaks the peptide bond to release the dinitrophenyl amino acid complex in solution, this one. And hence you have free separation and analysis of derivatized amino acids, HPLC or TLC separation, as we looked in the previous slides, to compare it with the standard amino acid uh, movement, okay, on the TLC plate. Then there's another technique called Edman degradation, which is used for purifying proteins by systematically eliminating individual amino acid residues from the peptide's amino end. This one also from the amino end. So this was developed by Per Edman, and actually the thing came upon like it came up because this was potentially devised as a solution to reduce protein damage during hydrolysis. So instead of using harsh conditions, what Edman did, um, novel labeling was introduced. Labeling and cleaving approach was introduced by adding phenyl isothiocyanate, this residue here, a phenyl thiocarbamoyl derivative form with the N-terminal here. Okay. So cleavage of N-terminal occurs under mild acidic conditions here. After this adduct is formed, this is getting cleaved under mild acidic conditions. Like eventually after the cyclic compound is formed. So this process actually preserves the protein integrity, leaving behind two peptide sequence components. The procedure can be iterated for each residue, allowing for the sequential separation of individual residues without compromising the overall protein structure. So Edman degradation is useful because it does not really damage the protein. It allows sequencing of proteins to be done in lesser amount of time, and definitely Edman degradation can be done best if the composition of the amino acid is known. To determine the composition, what also happens is the peptide should be hydrolyzed, and you denature the protein, heat it, and add HCl for a longer time, and hence individual amino acids get separated, and you can actually then again do chromatography to check what type of amino acids are present, and then you dye it with ninhydrin reagent, what we saw earlier, and it will be giving off a purple color. Or you can eventually also take absorbance or OD value readings, absorbance or optical density readings in a spectrophotometer, measure the readings at 280 nanometers, and check the presence of the amount of protein or amino acids in there, and hence you can actually check the composition but not the exact sequence of the amino acids in this way. So then comes ninhydrin test, which is a chemical test to check whether a given analyte contains amines or alpha amino acids. And ninhydrin is added to a test solution of the analyte, and deep blue color or purple color indicates the presence of ammonia or primary or secondary amines or amino acids in the analyte. Okay. So this is how the reaction takes place. And larger proteins cannot be really sequenced by Edman sequencing because less than perfect efficiency of the method is there. So what also happens is a strategy called divide and conquer successfully cleaves the larger protein into smaller practical amino acid sequence lengths, and like by treating the very big polypeptide chains into smaller like with enzymes or proteases, it is cleaved into smaller fragments, and eventually you can then take up these different sequencing techniques to get the amino acid sequences. Okay. So in order to put together all the sequences of the different peptides, a method of overlapping peptides is used. The strategy of overlapping peptides, if you remember, so what happens is in case of Sanger sequencing and all these things, you had some kind of nucleotide present at the terminal position, which did not lead to further extension of the nucleotide chain, right? Now here what happens is, um, so a method of overlapping peptides is used. So suppose now that what happens is different types of enzymes which have different specificities. Now suppose if you talk about this particular word, chromatography. Okay. Now just imagine, this is just an example, that chromatography, this particular word, each letter of the word is an amino acid. Okay. And this is, each amino acid is forming this particular polypeptide chain called chromatography. Okay. Now what will happen is, suppose enzyme A or protease A cleaves at any region after R amino acid. Okay. So this is just a trivial example. Okay. And R amino acid. So eventually if you treat this particular polypeptide with protease A, then what will happen is it will cleave after in R residue. Okay. So it will cleave after this here, and it will cleave here, eventually generating three fragments, okay, of this particular polypeptide. Now you take up, suppose protease B, which cleaves after, suppose A residue. So eventually what will happen is you will have chroma, then you will have TOGRA and PHY fragments, right? So when you overlap these fragments, eventually you will finally be able to, from these overlapping fragments, you will have, suppose there's, you take up another third protease which cleaves after T or something. Okay. Now, so all these mix and match, you will have different kinds of overlapping residues, and when you actually put those together, you will be able to frame the full polypeptide sequence. Okay. So that's how this strategy of divide and conquer works. Okay. So this is what is being talked about. Like a second time, you use a different enzyme or chemical to cleave into different residues, and you have two different sets of amino acid sequences of this particular same protein, but at different points, cleavage points. So by comparing these sequences and examining for any overlap between the two, you can eventually deduce the sequence for the original protein. Okay. For example, trypsin can be used on the initial peptide to cleave at carboxyl side of R, arginine, and K lysine residues. So using trypsin to cleave the protein and sequencing them eventually with Edmond degradation yields many different individual results. Similarly, chymotrypsin will cleave on the carboxyl side of aromatic and other bulky nonpolar residues. And eventually, if you see the overlapping fragments, you will be able to deduce the full polypeptide chain. Now you can also look at only the C-terminal residues. So what happens there is you treat the protein with carboxypeptidase to release the C-terminal amino acid and test the solution in a time-dependent manner. And either you use a different protein cleavage reagent producing overlapping amino acid, or you know, in order to, sometimes you need to locate the disulfide bonds present in a protein sequence. So you can cleave the protein by trypsin with or without breaking the disulfide linkage. And amino acid sequence analysis of the fragments will actually provide the site of the disulfide bond. Okay. And then again, there is mass spectrometry, which actually gives charge by mass ratio of the proteins. And you, from that, you have different, like different fragments of the proteins which are ionized. And eventually you have the mass and structure of the proteins, and eventually you will have the full molecular weight of the protein. Mass spectrometry has many different kinds of techniques in there. So just those are a little complicated, but the thing is, it can have, depending on how the things are getting, what are the matrix and how the things are getting ionized, whether a matrix is used or whether only an ionizer is used. Depending on the ionization techniques, there are different kinds of mass spectrometric methods. However, in general, mass spectrometry actually fragments a large protein and ionizes them, and then gets the mass by charge ratios, and then characterizes the full molecular weight of the protein. Okay. Now let's look at few questions. So a reversible binding of a protein with its ligand will not include which of the following? If something is reversible, then it shouldn't be a covalent bond, right? Covalent bond is a proper uh, bond which can't be really broken, right? However, these hydrogen bonds, van der Waals interactions, electrostatic interactions, or salt bridges are actually, if these can actually take place if they are in a certain distance. Two residues are at a certain distance within each other, like there's a certain distance within two residues. So in that case, you will have these non-covalent interactions. So all these are reversible. However, covalent bond is not reversible. Now, a protein which was run on a native page gel, it was calculated to be having 180 kilodalton size and molecular weight of the same protein. Native conditions, it was 120 kilodalton. However, SDS page, we know that SDS is a detergent. It is a denaturing page. And in that particular condition, you have 30 kilodalton molecular weight. So that means in the native conditions, whatever conditions it exists in the native state, it will actually, the interactions, the non-covalent interactions are actually cleaved off, or kind of uh, broken, and hence it eventually finally gives off this monomeric form which is 30 kDa. So now if we have to calculate the oligomeric status of the protein, it will be 120, which is the native page form, divided by the denaturing form which is 30 kDa. So 120 by 30 is 4. So hence it exists as a quaternary structure of 4 units, okay, or tetramer. So correct answer will be option C. Now the pi or isoelectric point of a protein is 6.5, and which of the following buffers should be used so that the protein can bind to a cation exchange column for purification? So cation exchange column, that means it exchanges cation. So it binds to something which has positive charge. So what will bind to something which has positive charge? So the matrix itself has to be negatively charged, right? So if the protein has a isoelectric point of 6.5, then it should be positively charged for it to bind to this negatively charged matrix, right? So which kind of pH will eventually give this particular um, will render a positive charge to this protein? So anything which has to be positively charged, so the pH should be less than the pi value. Okay. So here we see out of all these four options, only 5.5, which is lesser than the pi value of the protein. Hence, if the pH is lesser than the pi value of the protein, that means there are more H plus ions, and H plus will NH2 will exist as NH3 plus, and CO minus will take up H plus from the surrounding medium, and hence the overall net charge on the amino acid will be positive. Okay. Now a mixture of four proteins with their charges in a phosphate buffer of 7.4 are as follows. A uh, A has nothing, B has +1, C has +2, D has -1. So the protein which will bind strongly to a cationic matrix, like positively charged matrix, will definitely be something that has a net negative charge on it. Okay. So that will be option D here, which has -1 as the net negative charge on it. So it will eventually bind onto a positively charged matrix. So all these, this binding to different kinds of matrix are also different types of chromatographic techniques actually to separate different kinds of protein molecules. Okay. An initiator for acrylamide polymerization in SDS page is, uh, so this is a polymerization agent. Okay. SDS is the detergent here. Bisacrylamide is the branching agent basically. Timate is a catalyst for polymerization. However, the initiator for polymerization is ammonium persulfate, which gives the radicals where eventually the acrylamide and bisacrylamide can start polymerizing. Okay. So the correct answer is APS. Now, which of the following components imparts a negative charge to a protein? It is the detergent. SDS or sodium dodecyl sulfate. pH of the stacking gel is 6.8, resulting in the protein being sandwiched between which two fronts? We looked at it. It is tris and glycine fronts, where glycine exists as a Zwitter ion. It is extremely slow, and definitely tris moves towards the negatively charged area, and Cl minus moves all the way very quickly towards the positively charged electrode. Okay, the correct answer will be tris and glycine. Now, in a Western blot experiment, the same one that we looked at, only the SDS page proteins are transferred onto a membrane, and then there is immunodetection using antibodies. Okay, so in that particular experiment, a certain protein, remove the name from here, just the question that is being asked is the certain protein is actually probed with a mouse primary antibody. Okay, so that antibody that directly binds to this particular protein is cultivated or cultured in mouse, developed in mouse. So the secondary HRP, like substrate conjugated protein or antibody, should be. Now what happens is, now suppose the secondary antibody recognizes the primary antibody, right? Now, for an antibody to recognize something as foreign or antigen, for that to do that, now suppose if I cultivate or culture or develop both the primary antibodies inside my body, then what will happen is it is from the same source, from the same serum. So it won't recognize something as foreign to it itself, right? So what will happen is, because from the same source it is coming from humans, suppose, so then this the primary antibody won't really be a foreign material for the secondary antibody, which has to bind really to this antigen. Okay, so the primary antibody actually acts as an antigen for the secondary antibody. So if the primary antibody has been developed in mouse, then uh, the secondary antibody should be anti-mouse, because it has to recognize the primary antibody as a foreign thing. So something which was developed in mouse, the for that to behave as antigen, so the secondary antibody should always be developed in a very different organism altogether. Okay, so it should always be anti-mouse. Whichever the organism it is developed in, whether it's rabbit or goat or anywhere else, but it should be anti-mouse. So that is what you should be looking at in such questions and the options given in. The correct answer will be horse, like it is this antibody, secondary antibody was developed inside or cultured inside horse. However, it is anti-mouse antibody. HRP is the substrate bound to it, which eventually will be acted upon by enzymes, thereby giving off a signal. In case of HRP, it is luminescent, chemiluminescent signal. Okay. So a UV visible spectrophotometer can, this is a very direct question, can measure what? This is absorbance. A protein in the native state exists as a tetramer. The molecular weight of the protein is found to be 48 kilodalton in denaturing conditions. So the native molecular weight is. So this is exactly kind of the opposite of the question we looked at before. So now here it already says that it exists as a tetramer. However, in the denaturing conditions where it exists as a monomer, it is 48 kilodalton. So then in the native conditions, when there are no disrupting forces, then it will exist as a tetramer only. So in monomer, molecular weight is 48. In tetrameric form, it will have 48 into 4, which is, I think, 192 kilodalton. Yes, it will be, the answer will be 192 kilodalton. It will exist as a 192 kilodalton unit in the native conditions. Okay. So, yeah, that's it for today's session. Thank you for joining, and um, thank you for bearing with me during that video presentation. Thank you so much, and we'll meet again on next Sunday. Thank you. I will end the meeting.